Good day. This is Tuta Monimus from Monimus Varsity Connect International. So let us explain this phylum Porifera. Some people like to call it spongy. So this is the only phylum which is there in the subkingdom of Parazoa. Parazoa meaning that you don't have true tissues. Now, I used to pass bio. The reason why I was passing bio is that I would call safe people with such names of Parazoa, Porifera, Annelida, Chodata. I would just save people. Blastula, Gastulation, money fighter when I dealing with plants. I'll just save people with such. So this phylum of Porifera or sponges, scientists they are saying it is evolutionary dead. So it never proceeded beyond. When you look at the structure which we talked about in the last video of Parazoa, Parazoan, Eumetazoan split, you may go and look about. You can go and look to such. We explained. We said that this Eumetazoa, Parazoa, Parazoa split, animals started as multicellularity. After that, some animals had tissues, some animals did not have tissues. These animals which did not have tissues, these are the Phylum Porifera. And the Phylum Porifera, these are the animals who don't have tissues. So there are animals which have tissues and animals who do not have tissues. But these animals which had tissues, they developed further into other set of animals, hence developing further. But these animals which do not have tissues, like the Porifera, they stopped developing that time. So it means these animals, they are the ancient animals to exist. When I say something is ancient, I mean they lived that development happened many years ago. Yes, today they might have mutated somehow or changed because the homo genes, they experienced two evolutions in history of animals. But the same spongia which you see today, it has the same characteristics and is the same spongia which took place 600 million years ago. But insects and other animals are not the same. Animals developed, especially those who have tissues. This lineage sometimes is referred to as evolutionary dead because it did not give rise to any other group. There is no any other group which developed from this phylum. So these are the sponges. I hope you can see the sponges. These are the sponges. All of these and all of those which are beneath, these are animals. These are sponges. So sponges, because they are parazoa, they have all characteristics of parazoa. Parazoa, these are animals which operate at cellular level of organization and do not have true tissues. This means that cells are specialized so that different cells perform different functions, but do not come together to form true tissues. The reason why we are calling them animals is because they integrate their activity. They help each other. Some animals will do that. Some animals will, some cells in spongy, with like the ostia, will allow things to get inside. Some cells like the osculum will allow things to get outside. You can see that the ostia depends on the osculum because the ostia enters things only. But the parasite, there's a part called the parasite. The parasite will do what? Will close and regulate the opening of the ostia. You can see that the parasite is regulating. It is regulating the opening of the ostia, meaning that the ostia depends on the parasite so that for it to open, and also depend on the parasite for it to close. There are other cells like the amoebocytes. There are some amoebocytes which lymphoamoebocytes. There are some amoebocytes which transfer there's some amebocytes which digest. So, but they are also specialized. Like the amebocytes shall be the amebocytes. You should know that the amebocytes are totipent. So totipent means that they change into shape, into any shape that they want. The amebocytes, they are totipent. But it's not like the amebocytes, they depend on the porocytes. The amebocytes, they depend on the chinocytes. The chinocytes on the what? On the collar cells. So they are interdependent to each other. But they're not tissues, no. They're just cells which are specialized, but they need each other. But not needing them into, they just need them for some activity. That's why we say that there is integration of cells. Their bodies are just sort of loose aggregation of different types of cells. This is the simplest cellular organization found in metazoans. The sponges, these are the simplest animal ever. These are the simplest animal ever. These are the simplest animal ever. So let us go into details. Let us go into details about these sponges. So all sponges are sessile. They don't move. They are attached into something. They are attached into deep down the sea. These are animals. The sponges, you see them in, you find them in the sea. So they are attached deep down the sea. You find them attached inside. Sponges are characterized by numerous pores, which give them their name. So they have many pores inside. You guess why they have many pores? They feed by diffusion. Everything, things should diffuse inside. So they have many openings, small, many, numerous openings, pores. They have numerous openings. So that's why we call them porifera. Pores. So many sponges are asymmetrical. They do not have body symmetry. Know these terms, but asymmetrical means that you don't have a body symmetry. You can't even divide them into equal parts, but few exhibit a radial symmetry. Draw a circle now and divide that circle into equal sectors. In network called sectors. That's radial symmetry. The more you don't have a symmetry, the more you are primitive, according to the colonial theory. The more those animals which are asymmetrical, they are primitive or they are ancient, followed by those animals which are radial symmetry. Those animals with a radial symmetry, this one is animals which are bilateral symmetry. They are considered the latest animals. They are the latest animals. Bilateral symmetry, they are the latest animals. So, sponge that is supported by a skeleton made up of protein, collagen, and spicules. There are different kinds of spicules. Others that are monoaxon, others that are triaxon, others that are tetraaxon, others that are hexaaxon. These are the spicules. And which may be calcareous or silarious. When something is calcareous, it is made of calcium. So the protein, collagen, and spicules can be made from calcium or can also be made from silicon. Silarious meaning is made of silicon. So these skeletal elements, the chinocytes and other cells, are embedded in a gelatinous matrix called the myxohyli or mesoglea. Mesoglea is the middle layer lining the middle layer lining a sponge, and the chanocytes, which are the collar cells, and also this, you will find them. You will find them. The Mr. Glare. We like to call it the Mr. Glare, or the galactinous matrix, or the galactinous matrix. I want you to pause here. Take close to 10 minutes or 20 minutes. Study this structure. You draw it and study where each part. The same way you in the last lectures of this biology, animal diversity, you studied about the paramecium, also the protozoans, the flagellates, the amoebas, which are the cyclodinas. You studied about them, you draw them. Here, I will expect you also to do what? To draw and know the structure. But nowadays, biology, you write online lesson, and you science students who don't have a skill of drawing using a computer, especially those one which are new into science. But when you are fourth year and all, you know how to draw using a computer, they may ask you to draw such. These parts, they link. You can see the chinocyte, the pinocyte, pinacocyte, porocyte, ostium, spicules. You can see also the amoebocytes, that we have the archaeocytes. These are for nutrients. We have the sclerocytes. This is secrete spicules. You have the spongocytes, those secrete spongin. You have the colencytes, they secrete collagen. You have the lymphocytes, they are highly mobile. And also have the osculum. But we are going to discuss on how these animals feed. Sponges are filter feeders. Water comes carrying other things, like carrying bacteria, suspended plankton, and other detritus particles. The water is going to enter through the numerous small pores called the ostia. So after the water enters the ostia, we should know that the ostia are surrounded by a donut-shaped cells called the porocytes, and the porocytes, that they open and close to control the water flow. So there are some simplest type of sponges which water flows directly into a open chamber called the spongiole. You may refer to other diagrams that we have drawn. And water leaves the spongiole by a large opening called the osculum. So the interior of the spongiole is lined with the flagellated cells called the chinocytes, or the collar cells. So I want you to get the picture here, that water with the carrying nutrients for the sponge enters through the ostia. The porocytes open and closes the ostia. So the chinocytes they line the interior means inner spongiole coil. So beating of the flagellum of the chinocytes creates current that force water through the spongy plumbing system. So what is going to happen there is that the chinocytes is going to beat. And if they beat, what do? They capture. The chinocytes, they capture food. They do what? They trap food. Mind you, they do not digest. They capture. Sometimes they can phagocytize the food, but they don't digest. After the food is trapped or captured by the chinocytes, which are the collar cells, the food is going to pass into a second cell, an amoebocyte, waiting in the myxohili for digestion. The food is going to pass into the cell, an amoebocyte, waiting in the mesohili for digestion. Amebocytes carry food to other cells that require nutrition. Amebocytes carry food to other cells that require nutrition. We say that there are 5 types of amebocytes. Some are they are for nutrition, others for digestion, others to make collagen, others to make spicule, and others for movement. So we can say that there are 20%. Amebocytes can turn into any other cells that may be required by a sponge. 20%. When a sponge requires a cell to transport the food, the amebocyte is going to turn on that cell. When a sponge requires a cell to digest, an amebocyte is going to turn. So amebocyte turns to any cells required by the body. So this will allow the growth and repair of the end reproduction, because the amebocytes are also a. So amebocytes are responsible for producing of a sponge skeleton, and the skeletal system is made of a flexible protein, fiber, and all this. Spicules are usually made of calcium carbonate or oxides of silicon. That's why you can say that spicules that can be calcareous, they can be silly, they can be silurious. They can be calcareous, they can be silurious. These are the 2 things which are there. Why are they important spicules? Because they are important in the classification of sponge because of the shape and the structure. They give a sponge a shape and the structure. So this is the diversity. This is the diversity of the sponge, Porifera diversity, and I'll not teach it today in this lecture. I'll teach it in the following lecture of Phylum Porifera diversity, and I hope that you will be in a position to do what you will be in a position to appreciate it. And make sure that after you watch any video of us, you take it, you plug it in your status. You take it and plug. This is a secret so that we can see how our videos are. We are explaining basics of biology, lecture notes to guide you, but I encourage you that after any video of us, you go to other videos of senior professors and you watch.